All right. So this is the second chapter that we are starting. And so the infectious cycle, what do we mean by that? We are just going to give you an overview of that what actually happens and how we study the viruses. That's basically the overview of that. So this is also an overview of the of the virology class. So what we do is that the virologists actually divide the infectious cycle into steps to facilitate their study, but there is really no such artificial boundaries. So what do we mean by that? That in your textbook picture or the notes picture that you will see, you will just only will not see these red dots in here. These are the things that I have added, but you have the same picture. So what you are seeing that there are steps like attachment and entry, which is attaching to either the plasma membrane receptor or something, or fusing with the plasma membrane. Then you have the translation into the ribosome. Of course, there is mRNA making and all those kind of things that we're going to talk about. Then you have genome replication, and then you have assembly and the release. But these are not extensively separated effects. They built into each other, and there is no artificial boundaries. So this is a continuous thing, and things actually happen not one after another. It actually basically happens in a kind of an overlapping, synchronous way you can think. What do we mean by that? That when the first set of viruses are being assembled, there are still some maybe undergoing genome replication and packaging. So things overlap. So they just are not discrete, that once the genome replication is completely done, then it will, and the protein synthesis is completely done, at that time, it will start assembling. It's not like that. So that's the overview of the infectious cycle that we are going to talk about. So what do we need? What do we need to study the infectious cycle? And this is bold and big. We need a live cell. And what kind of cell? And this is a very important concept. And make sure that if you have your notes in there, you can just actually underline or highlight those words to understand a couple of definitions. So we need a specific type of cell from the specific host the virus infects. This is very important. Specific type of cell from a specific host. And based on that, we have some definitions. And you will see some important definitions about those specific cells. So what are these? So first of all, I'm just reading this, but that will give you the idea. A susceptible cell actually has a functional receptor for a given virus. The cell may or may not be able to support viral replication. So I'm just talking about in the context of a SARS-CoV-2, the COVID-19 virus. So you all know that a susceptible cell is, say for an example, epithelial cells of your nose or the pneumocytes of your lungs. These are the cells which are susceptible, and these have a receptor which is an ACE2 receptor. Now, is your gut intestinal cells susceptible too? They do have an ACE2, so they may be or may not be. We still do not know. We are learning that. Do your red blood cells do have the ACE2 receptors? They don't have the red blood cells. So they will not be susceptible. They cannot go inside and enter that. Red blood cells actually do not replicate anything because they do not have the ribosomal machinery. So that doesn't come into play as well. But white blood cells, yeah, some of them may have ACE2 receptors, some of them may not. So, but a susceptible cell will have to have a functional receptor for a given virus. This will allow it to enter. And for some of the viruses, there could be many cells that are susceptible in the same host, many cells that are susceptible. On the other hand, there could be some very specific cells that could be susceptible for that. Say, for an example, I'm just giving that a reovirus or rotavirus. These are some of the viruses which cause gastrointestinal disorder. These actually attack your gastrointestinal epithelial cells. If it goes to your lungs, they do not have the functional receptor for that, and it doesn't do anything. So that's what the susceptible means. The resistant cell has no receptor. It may or may not be competent to support viral replication. So here it is also a very important concept. So first of all, resistant cell has no receptor. Say if we just talk about the CoV-2, SARS-CoV-2, if you do not have an ACE2 receptor, it probably cannot get into the cell. And maybe, I'm saying maybe because there are some of the other theories that are coming up as well, that how it can enter the cell without an ACE2 as well. So, but for all practical purposes, if you do not have if you have a cell which doesn't have an ACE2 receptor, that cell is resistant to SARS-CoV-2. Now, if you take the SARS-CoV-2 and use a vehicle to deliver it inside the cell. Again, I'm repeating, take a vehicle to deliver it inside the cell. And we can do this in the laboratory for our purposes. We can actually package the virus in a liposome, and liposome is a membrane vesicle. It's made up of a phospholipid, usually a monolayer of phospholipid. We can artificially package the virus in a cell culture environment, put the virus in a liposome, which is a membrane, phospholipid membrane. And if we add these liposomes to the cells, phospholipid melts in phospholipid bilayer, and that's how the virus without a receptor can be delivered into the cell. But just take home message that somehow with a vehicle, we have delivered the virus so that it can enter the cell without the receptor. This is done artificially. If we do that, that cell may not be competent to support viral replication, but may be competent. The cell may, if the cell can gain entry into the cell, it may support replication or may not support replication. So you have both the things that can happen. The next one is a permissive cell that has the capacity to replicate the virus. So it will, if the cell is inside, so a permissive cell line or type of cell, if the virus is inside the cell, it will replicate the virus. And again, this is the important part. It may or may not be susceptible. What it means? If a permissive cell is susceptible, that means it has to have a receptor. It may not be susceptible means that it does not have a receptor. So a permissive cell line could be susceptible, a permissive cell line could be resistant. So just make sure that you have this concept. I have written that clearly in your notes, so you should not have any problem. So the best type of cell that what you have, a susceptible and permissive. That means that this allows the virus to enter the cell by the receptor. Permissive means that cell allows the virus to grow in the cell, is the only cell that take up a virus particle and replicate it. And these are the natural cells that what you can think about, say for SARS-CoV-2, this is your nasal epithelial cells or any epithelial cells in your upper respiratory tract, including the pneumocytes and many other cells around the lungs, including epithelial cells as well. These are susceptible, although there are some replication has been seen in different other tissues as well. So these are some of the interesting idea and a very important slide. So what we did with the viruses over the period of time, that we actually grow the viruses classically, first could not be routinely propagated in the culture cells. So before 1950s, before 1949, we actually could not grow the viruses in the cultured cells. But in 1949, this huge discovery actually gave the three scientists, you will see that in your notes, a Nobel Prize in 1954. And after that, routinely, they could propagate polioviruses in the cell culture, and that just opened up the whole area of virology. But before that, the viruses were grown in the laboratory animals and embryonated chicken eggs. And nowadays, we can just grow viruses on plants, fungus, protists, and bacteria. Important thing is like plants and some of the recombinant influenza virus are being made in tobacco plants. So you just crush the tobacco plants and isolate the virus for vaccination purposes from the plants. And this has been made. And out of those, you can just identify all of them. And this is a very interesting model that I'm just specifically mentioning here. This is a ferret that you can see. So they do have a long snout, and most of the respiratory viruses, including the SARS, all influenza virus, respiratory syncytial virus, these are respiratory viruses, the SARS and the SARS-CoV-2, they are all being tested on this ferret model. And the ferret is extremely susceptible to SARS-CoV-2 as well. Dogs, cats, there are some infections that have been observed in there, but they compared to the ferrets, they are less susceptible. But we are still learning the effect of the SARS-CoV-2, the host range of that, but they are comparatively less susceptible. But there are every day there are papers coming out of that. All right, so now, other than that, we will come back to those over the period of time. Here, just I wanted to just talk about one thing that we're just not introducing the term. Sometimes when the virus actually do propagate in these animals, over the period of time, they mutate. And one thing I will repeat again, you will see that this is also another myth that is out there. The people just talk, the virus have mutated. Now remember that the viral replication machinery is faulty. And this is a survival strategy for the viruses. So no matter what, the DNA viruses actually do have a little bit robust replication strategies compared to the RNA viruses. But no matter what, each replication cycle of a virus, in every replication cycle, in every cell, some of these viruses are mutating. So when you see something in the media, some people are saying that this virus is mutating, that's a completely off topic, in the sense that that is a complete myth. That does not happen that way. What they are trying to say, that it has mutated in a certain way to cause some additional problem. But yeah, that's a special kind of mutation, you can say, or explain that. But mutation happens in every replication cycle, in every cell, every time. And you can study these mutations in the animal models. So what happens that you infect a chimpanzee or a gorilla or a mouse or cat at one point of time, and you isolate the virus, say 7 days after the infection. And then if the virus is not virulent enough to kill but make the sick, and you just collect the virus every 3-4 days, and you match their sequences, their DNA sequences or RNA sequences, and you will see that there is a huge number or mixture of viruses in there which suggest the mutations. And this is actually happening with the SARS-CoV-2. It started in China, and gradually there is a website you can see that it is evolving over the period of time. Now, your question is that if it is evolving over the time or mutating, what does that mean? At this point, what it means that it has not significantly changed in a certain way that a vaccine development could be a problem. Not at this point. But you can easily understand that sometimes this happens with the influenza virus. In some of the years, the influenza vaccine does not work. So influenza vaccine is usually a mixture of 2-3 vaccines. And what happens that at one point of time, the viruses, the scientists actually look at the circulating strains and make the vaccines against those. These are usually a mixture of 3 or 4 different strains. Sometimes the virus mutate late in the season, and before that, the vaccines are already in the market. And at that time, that vaccine will not work. So this is a possibility, just to have a bigger picture of that. This is not necessary for this chapter, but you will have an idea that when we routinely grow the viruses in the animal cell culture, we also take care of, or we study their mutations as well. And we'll talk about that in a separate chapter. Now, Viruses are routinely cultivated in cell lines, and what you are seeing that there is a difference between the cell lines, and this is from your cell biology. You may or may not have taken it, but the differences in between the cell lines is that there are some cell lines which are called the primary cell lines. So the primary cell lines are isolated cell from an organ tissue, and we just chop them up and break them up with the proteases so that they're not attached. And after that, we just propagate them. These are good for 520 cell division cycle that we can just transfer them from one vessel to the other, grow them for 520 replication cycles, sometimes maybe 50. There are a few exceptions that you may do it for up to 100 times. But after that, they will basically die off. Why we use then this primary cell culture? The reason for that, these primary cells are most naturally occurring cell types for a viral replication. And that's the reason that why we use these primary cell lines. So this is, say, for example, from a human foreskin fibroblast. This is a primary cell line. Now, why we use the other type of cell line and what it is, there are other type of cell lines right here, which we call as an immortal cell line or a transformed cell line. What it means that there is something happened to their DNA. Most of their has an aberrant chromosome number, which are called aneuploid. They are not diploid. Odd number of chromosome, many mutations. And these are basically, think about that, usually an oncogenic tumor or a cancerous tumor have these transformed cells. Once you have that, these cells actually get immortality and they will grow forever. So these are extremely useful for the scientists to grow the viruses, because you do not have to make these primary cell cultures every 5 or 20 generations after that. This will grow forever. Why we sometimes cannot use these cell lines, although they are routinely used, because they actually lose their native physiological conditions, and that may not be the exact replica of a cell line or a native cell line which is present in an organism like a human being. That's one of the problems. Second problem is that sometimes the viruses, vaccines are made in some of the cell lines. The vaccines are never made into these cell lines, which are these transformed cell lines, which may, if by chance some of these oncogenic DNA is isolated along with the viral particle that you are trying to generate the vaccine from, the vaccine may contain some of the DNAs which could be oncogenic. So when you inject, there is a possibility that some of these oncogene can be delivered to the patients and the patient can develop a tumor. So that's a huge problem, and that's the reason no vaccine is done on these continuous cell lines, which are does have these problems. Now, here is a cell line that you will read in your notes as well, which is called the V38, and this is derived from a human embryonic lung. This is a diploid cell strain, and this can be passage up to 100 times. And this is not a continuous cell line. It does have a mortality, but this is used for making vaccines. So this is a very popular choice for making even poliovirus vaccine. Much more, they live much more longer than normal, usually the normal other primary cell cultures. So that's the overview of your virus cultivation. Now, sometimes, just another thing, that sometimes the scientists need viruses in a very large amount. Say, for an example, when the structural biologists need the viruses, particularly for their extracrystallography studies and those kind of things, they need the viruses which could be weight-wise, they need milligrams of these proteins and viruses. So sometimes some of these cells which actually grow in a monolayer means that you take a vessel or a flask and they grow in a single layer on that. So here, what we are looking at, these are the fibroblasts, and you can see that these are the single layers of cells that are growing. So you have a limitation of growing. So you have to grow hundreds of flasks to have enough amount of virus for your experiment. So what you do, that there are some cells which can grow in a suspension culture. That means that they are not adherent to the surface. Say, for example, these fibroblasts, they need an attachment like this plastic to grow. On the other hand, there are some of the insect cells, some of the lymphocytes. They can be used in a culture like a flask, which has a magnetic star bar, and you can have a very high concentration of cell, which can produce a large amount of virus in the laboratory. You can just easily go and look at some of these pictures online, that how these cell cultures are. You will be able to see that. So those are another application for growing these viruses. All right, another very popular idea is growing the viruses in embryonated chicken eggs. So these the eggs are fertilized, and then then they are inoculated with the viruses, and you can see that there are different viruses in different areas. You have a chorioelanthonic membrane inoculation just at the very surface. Then this is the amniotic inoculation just close to the embryo. This is in the yolk sac, and then you have an allantoic membrane. And the thing is that all of these do have different types of cells around them, and these cells actually do allow the virus to replicate in those areas. And here you can see a list of viruses which grow in certain areas. You do not have to memorize those. Like, I will not ask that which of the following virus is growing in chorioelanthonic membrane inoculation. I will never ask that type of questions. So it is just more of a conceptual idea, but you do not have to memorize this at all. All right, so here you are seeing that how the chicken eggs are actually done. So after inoculation, as I said, that after a few days, there is at the very top, it drills a hole and inoculates at a certain way, and this is completely used by the robots, and it takes less than 10 seconds to inoculate a row of days. So eggs at 10, 12 days being inoculated by an automated machinery. So it's like your influenza vaccines are still mostly made in this embryonated chicken eggs. That's how the industry actually does that still today. All right. So now the cytopathic effect. What happens to the cell once they are infected with the viruses? And these are called the effect of the viruses on the cell, and a cytopathic effect, or the pathology of the cell. So what we are seeing, that this is actually a cell which is in a monolayer. So this monolayer of cells, you can easily see that they are attached to each other right here. Now we have inoculated them with a virus, and you can see now that there are some cytopathic effects. Some of the cells are rounding up more, they're rounding up. Many of them have completely lysed, and the complete destruction of these cells. And you can see that this is just basically a phase contrast microscope, light microscope, where you can see these cytopathic effects. There are many different types of cytopathic effect you can easily see. Here is another idea that how the cytopathic effects are. So if you carefully see that, if you look at this cell right here, you can easily see you have a nucleus right here, you have a nucleus right here, and it's a giant cell. And it is a giant cell means that a syncytia is a fusion of multiple cells. So what happens, that suppose this is a single cell, it's fused into two, and multiple cells can fuse together and form a syncytium, which is a singular, and syncytia is a plural. So why this happens? So this is a strategy for the viruses, which actually it does. Like a Sendai virus is one of the virus which actually does this fusion or the syncytia formation. In this large cell that what you are seeing, this becomes a viral factory. So basically the hundreds and thousands of viruses are being made in this factory. It just got a bigger space, basically, rather than on small individual cells, which will die very soon. they are actually allowing this giant cell to live a little bit longer and making use of the space before killing the virus. And this is called a syncytium formation, and this is a typical uh, cytopathic effect. And there are several other type of cytopathic effect, and this is very self-explanatory that you are seeing. You can just read the list right here. And again, you do not have to memorize each specific morphological alterations and the viruses associated with that. Just know that what are the kind of cytopathic effects are, like nuclear shrinking, proliferation of nuclear membrane, vacuoles. Sometimes you can have virions in the nucleus that you can see Negri bodies. So this one is one of the important things that you probably have heard in a microbiology class as well. So this one is one of those that you probably want to remember. That's just the only one. So Negri body is basically some of the specific dark spots in the cell which is found in the rabies virus. These are the virions in the cytoplasm. These are the viral factory. So just only this line. And this is the virions in the cytoplasm, rabies virus. That's it. The others, you do not have to specifically remember that what it does. This is just only, we just talked about this Negri bodies. That's why that. And you can easily see the coronaviruses also do the syncytium or the cell fusion formation as well. One of the things that I just want to mention, that because it is important right here, which I did not mention, this is in human epithelial cells, which is called the HeLa cells. And these HeLa cells was actually obtained in 1950s from an African-American lady who went to Johns Hopkins University for the treatment of a cervical cancer. And her name was Henrietta Locklear. And from her, this cell was isolated in the 1950s, and this is the cell that is actually used the highest amount of cells of all that is being used in the whole world. And you can easily imagine the cell line which actually was taken without her knowledge or anything like that. At that time, it was it is totally unethical at this point of time. And that has been used for so many years. So there is a book by Rebecca Scoot, and I just tell that in my cell biology class as well, An Immortal Life of Henrietta Locklear. So just definitely, if you have a chance, read that book by Rebecca Scoot in there. So it's a very, very interesting story. All right. So I will just put that in the note. It's Rebecca Scoot, Immortal Life of Henrietta Lacks. Then the next part is the assay of viruses. The important definition is the viral or the virus assay is a method or process of measurement of a virus in a sample. And the method could be physical, seeing through a microscope, or biological infectivity. And another definition, viral or virus titer, is the measurement of concentration of a virus in the sample. Nowadays, as we are doing these coronavirus measurement, people actually, or some other, you have definitely heard that you actually look at antibody titer. And antibody titer means that you are the measurement of a specific type of antibody, how much concentration of that antibody is in your serum from a blood sample. The same way the concentration or measurement of the virus is a viral titer. So first, we are going to look at some of these biological methods, and there are some of these assays that we are going to talk about. The first one is the plaque assay, and Renato Del Becco in 1952 developed this plaque assay for animal viruses and received a Nobel Prize for that. And this actually revolutionized the way we actually assay viruses, an extremely powerful technique, and every virology lab actually does that if their viruses forms a plaque that we're going to talk about. So what is a plaque assay? Just an overview of that you take a Petri plate, and on that Petri plate, you plate the cells, and they will, suppose they are growing in a monolayer. Now, remember that what we mean by a monolayer, this is an important concept, just if you are just not familiar with that, think about that this is the bottom of the Petri plate, and the cells are growing in just one layer, not on top of each other. So each one of these things that is growing on the bottom, and this is your monolayer. So I can use my pad whiteboard, but not necessary. So this is your monolayer. So that's growing in here. So what you do with this, after you infect it with a certain amount of viruses, it will infect a cell, and this cell will eventually die. And once it dies, it will release the viruses, and it will infect the surrounding cells. Now, the surrounding cells will die, and the viruses are going to come out. And it's basically this is killing a region of the monolayer. And this forms a clear zone, which is a plaque. So it actually can be visualized in a phase contrast microscope. Right here, you can see that monolayer, which is destroyed here. Sometimes what happens, the virus is inserted with a chromogenic substance, is inserted with a gene, which actually do produce an enzyme. And when you add a chromogenic substance, it does a reaction to produce a blue color as well. And these are usually used for mainly vector preparation or recombinant DNA technology as well. But without even that, how you actually can visualize some of the pictures that you will be able to see right here, you can. So here you can see that this is a monolayer. And look at the middle portion of the area. There are some green cells, which is the virus is expressing those proteins which are green in color. Now you can see that the plaque is actually growing and growing and growing, and that area of monolayer is become cleared up. And you can see that the viruses, as they are expressing that green fluorescent protein, these cells actually do have those green fluorescences as well. So the green fluorescence protein is just a reporter protein that is present in those cells. So isn't that amazing that how a plaque is formed? And this is basically the plaque is formed in a cell. All right, so how we do this process? This is a process that this is very similar to a serial dilution assay that you have heard in your microbiology. So you take a viral stock and then you serially dilute it. So you just take, say, 0.1 ml to 0.1 ml for the virus. Probably you have done this in your may or may not have done that in the microbiology. Sometimes you take 1 ml of your E. coli, add it to 9 ml of E. coli. Doesn't matter, but just 1:10 dilution. So as you are taking one tube to the other, each time you are diluted the virus here 10 times. So you can see that you have the dilutions at 10 to the power -1 all the way up to 10 to the power -7. Here, there are too much viruses. We actually do not take these. We usually take these higher concentration. And from there, what we do, that we take just 0.1 ml, 0.1 ml, 0.1 ml in some of the Petri plates, which does have 90% confluent cells. What we mean by confluent cells, the confluent cell means 90% of the Petri plate, 90%, not 100%, then the cells will start dying before the virus actually replicates due to the contact inhibitions. Because some of these cells which grow in monolayer, if they touch each other, they will stop growing and then they will start dying because they cannot pile up on each other. So you do not want 100%, but about 90%, so that the virus does have time to replicate in them. And confluency means that 90% of the Petri plate surface is covered by the cells. You have just 10% space. By the time the virus actually starts replicating and produce the plaques, it will cover 100% of it. So you inoculate that, and then the virus 0.1 ml, and usually you try to absorb, or which is basically the virus binds to the receptors of the cell, incubate, and then remove the inoculum. We do not leave that. And after removing that, we actually overlay, this is the important part, we actually overlay with the agar, that's common, or the CM cellulose. These are nothing but the same thing as a gelling agent. So once we cover these, all of them, we actually incubate them to 48 hours up to 2 weeks, depending upon the virulence of the virus. Some of them form plaques very fast, some of them takes much time. Then what we do that we actually stain these cells with usually crystal violet and then count the plaques. And from those, you can easily find out the plaques. Usually we count plaques in between 10 to 100. Again, 100 is also too much. We tend to count in between 10 to 30 plaques per plate, which is statistically, the data is very good for that. So suppose we found here in this plate 17 plaques. So now we can back calculate the total number of a virus in the sample from here. How? See that right here, we have taken the dilution that we took is 10 to the power minus 6. We have taken okay. I will answer your question, Cooper. Yeah. So right here, what we are looking at, that when you get this 10 to the power minus 6 and you have taken 0.1 ml, so here the dilution factor, it becomes 17 into 10 to the power 7, or 1.7 into 10 to the power 8. And these are called the plaque forming units per ml. So this is how you actually assay the virus or amount of virus in your stock. So here, what it is saying that Cooper asked a question that in the notes it says plaque assay is used mainly to determine the titer of the virus. So you want to know the titer or concentration of the virus in this. And this is how I found the titer right here by back calculating that in this, there are 1.7 into 10 to the power 8 plaque forming units per ml. That's my virus concentration. Now, what do we mean by a plaque forming units? And I'm going to explain that in a second. The reason for that, we use the word plaque forming unit. The virus particles present in a sample does not mean that every virus particle is in a perfect shape and capable of infection. Sometimes what happens that many of the virus particles are defective and they will not be able to enter the cell and replicate. So instead of telling the 1.7 10 to the power 8 viruses per ml, we call it 1.7 into 10 to the power 8 plaque forming units per ml, which represent these are the number of infectious virus particle or virions that we talked about in the first class in that sample. Also, another second part of Cooper's question, does this method also give the scientists a rough idea of the life cycle of the virus? No, not really. For the life cycle of the virus, what it does, we have to employ different steps and methods that we are going to talk about in mainly after the genetics, say how it replicates in a cell, how it translates, how it packages, and those will be the subsequent chapters. This is basically a method to determine the concentration, to start an experiment, to study those subsequently. So this gives you just the idea that I have a cell line, I have to infect those cells to produce the viruses. If I infect them with too many viruses, I will not get a good production of the viruses because they will kill them beforehand. If we have too less, then it will take too much time. So we need the right amount of viruses. And then Matthew asked does a plaque form only one viral particle? Yes, theoretically it is. It is formed from one infectious viral particle. There are some exceptions. We'll come to that in a second, which is called a two-hit kinetics. So we'll does that towards the end of this chapter, where most of the time it will be just one infectious viral particle going into one cell. but things can get complicated if theoretically two of them get in there, and we will have a different kinetics. But for most of the practical purposes, one plaque from one viral particle. That is correct. So right here, that what you were talking about, if one virus particle gets into one cell, forms a plaque, what we get is called a one-hit kinetic. Number of plaque is directly proportional to the first power of concentration of the virus inoculated. If the concentration of the virus is double, the number of plaques double. So you can check that from that. So suppose from this experiment, you got the viral titer, and you double check this experiment that whether it works like that as well. So you now can determine the number of cells, and once you have the concentration, you can inoculate them, and you will be able to predict the number of plaques that is being formed. If it follows this one-hit kinetics, then your calculation is almost correct. Sometimes you can get a two-hit kinetic, and this is a number of plaques is directly proportional to the square of the concentration of the virus inoculated. Now, here is a complicated way that most of the purposes, this is a small amount of cells can actually have that. And that statistically, there could be a little bit of error, but that takes care of that. But there are some exceptions that there are certain viruses, some RNA viruses, which have two copies of the genome. They are separately packaged inside the virus, and then the virus particle is actually formed. And they show a very different kinetics, which is this parabolic kinetics, which we call as a two-hit kinetics. So you have to have different types of plaque assay results from those two-hit kinetics, which are some very specific viruses. And we are going to talk about this much later as well. The second advantage of this plaque, that what you are seeing, that for most of the purposes, we assume that one virus is coming from, one virus is infecting a cell and producing one plaque in all theoretical respects. To do this, what we do also, we do what is called a plaque purification. So we take this one single plaque, and then we actually, this is just sucked up just by a micropipette, like a 1 ml tip micropipette, and this will be an agar plug. We just put it into a media, and then this will have the virus from here. And then with this, we will do another plaque assay. We will, from that plaque assay, we will again take the virus from that, and we usually do it 2 or 3 times. So once you have that, you assume that at least for the second or the third time when you're doing the same thing, chances of 2 different viruses getting into the cell is much, much less. That's the reason that we do 2, 3 times. So from here, after 3 times, usually when you get that, usually you should have a clonal virus stock. That means that in that sample, all of the virus should have almost the same genetic makeup. So this is called what we call as a clonal virus stock, that what is being used. So these are the 2 different purposes of the plaque assay. One is for the titer or the measurement of the virus, calculation of the virus, and plaque purification to make a clonal virus stock. So that's it. So other than that, there are some very quick methods. Some of these processes or the plaque are not sometimes formed in some of the viruses. They just don't form plaques by killing the cells, because they're kill the cells much more slowly. It doesn't form plaque. So you can do a fluorescent focus assay for that. And this is the identification of viral focus by immunofluorescence microscopy. So either you can add an antibody tagged with a fluorophore, or you can have a virus which is expressing a protein like a green fluorescent protein that you have seen in that video. And then you, under a fluorescence microscope, you look at these fluorescent focus or foci. So each one of these dots is one foci, and you just count them the same way. And you can see that this is an overlay from a phase contrast microscope. You can see the cells in the background, and these are the fluorescent focuses. And you can just measurement the dilution that's the same way. And instead of a plaque, you just assay these. That's one of the modification. The same way, another process is called an infectious focus assay. And this is a modification of plaque assay. This is how exactly a plaque assay actually look like. And it looks like a plaque assay. But the modification of the infectious focus assay is mainly used to identify, right here, the second, this is important, to identify the fraction of cells in a culture that are infected with a virus. So what you do, that you make these monolayers like this in all cells, and you take some of the cells from a different culture and dilute them. You dilute the cells, not the virus. Remember that. You dilute the cells from a different culture, which is infected with the virus, and add those cells onto this monolayer of cells. And look at that, what number of cells or percentage of cells are infected with the virus. No number of cells, not virus, are taken to infect uninfected susceptible cell monolayer to form the plaques. And here is the last one, is a transformation assay. Some of the viruses actually do form this transform focuses. What do we mean by that? What you are seeing right here, this is one of the transform focus. And this is mainly done by some of the oncogenic viruses or transforming viruses. What you are seeing in the background, these are the cells in the monolayer, because what happens, these cells will have a contact inhibition, so they cannot pile up against each other. When you infect with them with some of these transforming viruses, like for example, sarcoma virus or RSV, they actually make these genetic changes into the cell. They can become oncogenic cells or transformed cells. At that time, they lose the property of the contact inhibition, and they can pile up against each other. And again, remember that instead of that, we drew that picture beforehand. So now they will grow on top of each other rather than monolayer. Here is your monolayer, but here, so instead of that, so this will be your transformed assay, which is essentially piling up of cell, and these are very, very nicely visible. You can see that there is one focus, one focus, one focus. This is just like the plaque assay. So you know that this focus is formed by one virus, another virus, another virus, and it's the same way that you actually measure the viral titer the same way. And there are many other ways we can do that. The endpoint dilution assay, and we'll stop on this one, is basically what you do, that you just plate the viruses with the cells in these multiple boil plates and with the different dilution, and you look at the cytopathic effect. Why you do this experiment? To look at the cytopathic effect, because some of the viruses will not show any of these. You can only see the cells are clumping up. So you are looking at these cytopathic effect, and what you are seeing, that at the 10 to the power -5 dilution, 50% of the wells, like see right here, 1, 2, 3, 4, 5, and you have 1, 2, 3, 4, 5. So approximately 5 positive and 5 negative. So 50% of the cells showing cytopathic effect, and this is just by looking at the cells under the light microscope, and you can tell that here the tissue culture infectious dose of that virus is 50%, or TCID 50%. This is also another way, which is called that endpoint dilution assay. And you can see that right here, almost you do not see any cytopathic effect, not in the 10 to the power -7. On the other hand, you see cytopathic effect in all the cells. So here you are just measuring which concentration of the virus can infect 50% cells in the culture. So we will stop in this one, and we will talk about this particle to PFU ratio that we have talked before. All right, so any specific question that you have? Pretty much we stopped at this slide, which is the endpoint dilution. And what endpoint dilution, I'm just trying to just give you another idea about this, that here you are seeing a 96 well plate, and the endpoint dilution assay really doesn't have to do you do not have to do this experiment in a multi-well plate like this. You can just do it in an animal directly. Like you can just have macaques, like monkeys, chickens, or mice, rabbit, ferret, any animal model. And suppose you have a population of sometimes these animal models in 50s, hundreds as such, and you dilute the virus and you just in somehow intranasally by injection or somehow you just depending upon that what kind of virus, usually the respiratory virus, just give them an intranasal dose, and then you just wait and see that after a certain period of time, after 5 days, 7 days, whatever the incubation period, you check those animals and you see that how many of them are sick, and you determine that what dose of a virus went to that specific animal. And when you just do the sum of many replicates, pretty much, you just find the endpoint dilution assay. Whereas here, what you do that you just plate equal number of cells in each of these well, and you dilute your stock and add the virus to each of the well, depending upon this serial dilution. And then you just look at them under the microscope for cytopathic effect. And if you're looking at the cells that only show cytopathic effect, you can do that in the cell assay. And here you can find that equal number of cells are negative and equal number of positive. So here we find this as a tissue culture infectious dose, 50% we call it. And the virus stock that you have is at 10 to the power -5. So this endpoint dilution assay was actually developed even before plaque assay, because some of the viruses actually don't form plaques that you know, and there are various variations that you can do with this assay. So next, what is very important is the particle to PFU ratio. So what we mean by this particle here we are talking about is the number of physical viral particle. And the virion, when we define the virion, a virion is an infectious virus particle. Now, when you have this mixture of, when you have a virus stock, does not mean that if you have 1 million virus in your stock, that 1 million of those virus particles are infectious. There could be many different things that due to the reason that they're not infectious. So the particle to PFU ratio is the number of physical particles, viral particles, over number of infectious particle. And we express that as a plaque forming unit. And we know that what it is, because we just, for theoretical purposes, we always calculate that one virus forms one plaque. Yeah, of course, we just agreed to that if two viruses simultaneously do that. But for most of the practical purposes, one infectious particle will form one plaque. And that is very important. And it is a very interesting thing. For each of the viruses, there is a certain particle to PFU ratio. What we mean by that, say right here, for the papillomaviruses, it is 10,000. What we mean by that when you are culturing papillomavirus, or when you are infecting an animal with a papillomavirus and you have a viral stock, in that viral stock, a large number of particles are defective and they are not infectious. So this is actually very interesting, that a large number of them are not, oh, no, you do not have to memorize this. Yeah, so one thing is that I will just put that in the notes underneath the slides, that you don't have to, like dates, which year what was discovered. You do not have to memorize any of those. I mean, like, just you guys are seniors, just have your overview of that any line by line memorization or numbers or something like that, I will never ask those numbers at all or something like that. The concept is more important, but I will just put those in your notes that you do not have to memorize those at all. So this is just establishing the point and just giving you the point that how it varies among the viruses. That concept is the important one. For some of them, you can see that it is very, very large. On the other hand, if you just look at this is an alpha virus, which is representative, is a Semlicky forest virus, whereas the particle to PFU ratio is almost 1. You cannot have a value of particle to PFU ratio less than 1. That is the lowest number that is possible. That means that if you have a 1 million Semlicky forest virus particle, all of them, the million of them, all of them are infectious. Or maybe if it is 2, then 500,000, 500,000 of them are infectious, and which is an astronomical value for most of the viruses. You can see it's like 1 or 2. So Semlicky forest virus is, and it just depends that how, we will just discuss that what determines the particle to PFU ratio. So you can see that SARS, this is the classic SARS. This is not SARS-CoV-2. It is in between 67 and 540, and the average, that number that you get for the classic SARS is about 240 particle to PFU ratio. So of course, your question will be why I did not put SARS-CoV-2 in there, because this is being determined yet. And we do not know this number because there are different types of cell studies where when the different laboratories are making their viral stocks and studying them on different cell line, like an E6 cell line, Vero cell line, these are the popular cell lines that they're using, and they are seeing different doses. And at this point, we do not have this number, but definitely this number will come out soon. So if you see the SARS-CoV-2 particle PFU ratio in publications, you will see that there is a varied number of those. So that is important. So why this actually happens? So the number of physical particles over the infectious particle gives you the number. And here what happens, if it is 1, a single particle can initiate the infection. On the other hand, think about that if that number is 10,000, then just to infect one cell, theoretically, you have to give them 10,000 virus particles. So your dose is much more if the particle to PFU ratio is very high. This concept is important. The dose is much more. If it is high, the dose of the virus will be less, or the infectious rate will be much less, because the virus is infectious in a very small dose because of the low particle to PFU ratio. That's the important concept that you have to master here. And why it is so? Not all viruses are successful, and they damage the particle. There are constant mutations, and there are lots of complexity of the infectious cycle, where some of the viruses cannot produce a large amount of successful viral particles which are infectious. And of course, this complicates the study. And that's the reason that we do not have these numbers for SARS-CoV-2 yet, already 6-8 months into the pandemic, and we do not know this number because it is still complicated. Different cells giving you different numbers, different animals giving you different numbers, so you exactly do not know. We know these numbers for the macaques or the monkeys and the ferrets or something like that, but that number does not have any relationship with the human beings. These numbers will come up gradually, but it will take much longer. Large number of pools are required. So a higher PFU equals to lower infectious rate. So the thing is that what we mean by the higher PFU, lower infectious rate is so at the same dose, it will be a lower infectious rate. What we mean by that, let's take a look at this. This is a good question. That, say, for an example, if we take 10,000 papillomavirus particle, theoretically, and this is very crudely, I'm saying this is all in cell culture, but what I'm saying that if you take 10,000 papillomavirus, technically, it can form one plaque forming unit, and think about that one person can be infected if you give them 10,000 virus particle. Whereas for the Semlicky forest virus, if you have one virus, you can infect one person. So lower the number, usually the infectious dose is is much higher. So lower the particle to PFE ratio, infectious dose is much lower in the sense that you do not need that many virus particle in there. And you can just correlate that. Most of these studies are in the cell culture. So you have to, and why I just put complicates the study, this study exactly does not correlate with what you see in the physiological condition. And say papillomavirus, this is the route is the reproductive tract, and you have the mucous membrane and all those kind of things. On the other hand, if you just look at the herpes viruses, the skin sometimes is the route for those. On the other hand, for the influenza virus, you have the nose or the mouth, intranasal or mouth is the route, and each of these what we call as a portals of entry for the virus are protected by different things, and really you cannot compare apples to oranges. So what do we mean by that? If you are taking the say orthomyxovirus, which is the influenza virus, you take 50 of them or say 10,000 of them, and you just put them in the nose, you will see a very drastic difference if you just put 10,000 virus particles in a different route. So those complicates the study overall. But mainly what you are looking at is in the cell culture experiment particle to PFE ratio. But roughly you can have an idea is that higher the PFE ratio, that as Karan actually said, that higher the PFE ratio, lower is the infectious rate. But we will use this term a little bit judiciously or cautiously because it is a little bit more complicated just rather than the plain numbers. And the mutations, we are going to talk about that every replication cycle, lots of these viruses actually constantly mutate, and some of them mutate into viral particle, which will not be infectious again. And here we will completely bust the myth that you hear all the time that virus has mutated. You will learn gradually somewhere else that every replication cycle in every cell, these viruses constantly mutate. And some of them will mutate into virus particles, which will be completely damaged in the sense that they will not be able to surmount an infection as such. Some of them could be attenuated. That means that some of them can form an infection rate where you have a particle to PFE ratio becomes much higher. The ratio becomes higher compared to that. And these complicates the study. All right. So next, we are looking at some of the viral assay method, physical methods, and these are very straightforward. So let's take a look at them one at a time. And the first one, these are very self-explanatory. You have the electron microscopy. So when you are doing the transmission electron microscopy or cryo-electron microscopy, you just use a negative stain, which gives you a contrast. Here you are seeing a classic bacteriophage with a negative stain, and this is a transmission micrograph of that. The scanning electron microscope actually will show only the surface feature of the virus, where the transmission electron microscope, you can see the full feature of the virus or inside the cell of virus when you take a section of that. These are all from your different courses in, say, for an example, in microbiology, cell biology, you have learned about this. There's really not detail. We do not need that. And then you have the fluorescence light microscope. And again, the fluorescence light microscope can detect the virus with the light microscopy. But if you just add a fluorophore to the virus only, because a single virus particle, except the giant viruses that we talked about, like a Pandora virus or the Mimi viruses, and these are really large viruses which are visible with the regular light microscope, even 1.5 micron. But most of the viruses, 99.9% of the viruses, are too small to see with a regular light microscopy, but you can see them if they're tagged with a fluorophore. Sometimes you have recombinant viruses for many types where you have a green fluorescent protein or a red fluorescent protein. is tagged to one of these or fused to one of these proteins. These are the recombinant viruses. And you can look at the single virus particle here, assuming that the green ones are the virus particle. And you can visualize them with a regular microscopy or confocal. But when you had the super resolution fluorescence microscopy, you can literally see single viral particle moving along the actin or the microtubules, and you can track them actually. So very interesting studies can be done with the super resolution fluorescence microscopy. All right, after that, we are looking at a classic assay, and this is called the hemagglutination assay. And these are done with many of the viruses which actually cause this hemagglutination, what I mean by that, that these cause red blood cells to clump. And that is being shown here. So what is exactly what is being done? So for an example, I'm just giving a classic example, and the picture is the same thing is here. This is a typical influenza virus. And the influenza virus actually binds to a glycoprotein, which is the sialic acid, and it binds with an N-acetyl neuraminidase acid is responsible with that glycoprotein to bind to the sialic acid receptor. Now, this N-acetyl, this detail is not necessary, but just the overview of that. And due to that receptor interaction with this virus, they can actually cross-link each other. So that's the important part. So how it is? So on this virus particle, you have this N-acetyl neuraminidase acid. It will attach some of these glycoproteins to the receptor on this red blood cells with its sialic acid, which is present on the red blood cell. And some of the receptors of this red blood cell will attach to some of them, and they will form this cross lattice, and that cause this hemagglutination. And we can observe some interesting stuff in that. So what we are seeing right here, these are the virus dilutions. You can see that the different 1:4, 1:8 to all the way to this. And the A wells did not receive, this is a control, did not receive any virus particle. And what you are seeing, what we call as a button or a red dot at the bottom of the well. So what happens when you don't have any virus added to the red blood cells, all these red blood cells will sink at the bottom of the well, and they will form this characteristic dot, red dot, which we call a button. But the second, which is the B line, the lane B or the row B, you can see that as we are what you are seeing that we are not seeing, and I will come to this later, 1:4. But as we are seeing that in these dilutions right here, pretty much we are not seeing any of these buttons, but we are started to seeing buttons at 1:1024, 2048, 4096, 8192. We are seeing the buttons. So here, the hemagglutination is not happening, whereas here hemagglutination is happening. So when the hemagglutination actually happens and the virus forms this lattice, these lattice formation doesn't allow these red blood cells to sink at the bottom, and you don't see these buttons. So what is happening right here? So this is an interesting thing that 1:4, which is a very high viral dilution, the virus actually does have an enzyme which is called neuraminidase. And this is the N that you see all the time. This neuraminidase actually cleaves these N-acetyl neuramic acid, and what it does, that N-acetyl neuramic acid actually, so once it cleaves it, that will not allow the virus to attach to these red blood cells, and they will all be separated. So the concentration of the neuraminidase is too high, and that cleaves that N-acetyl neuramic acid, whereas when you dilute these, you do not have that concentration to cause that. And that's the reason that this is what we have. This is what you are seeing. You will see that in the note, it's not needed that detail, but that's the reason that at a low virus concentration, due to the high amount of neuraminidase, we see this button, but we don't see that. So pretty much here, the dilution, you can see that 1:5, 1:12, that's probably the well, which is the up to which you can see the hemagglutination. So this is where you don't see anymore. So you will pick that number as the dose. All right, so that's your hemagglutination assay. And then you can just look at some viral enzymatic activity. And again, you do not have to explain any of these curves, just an overview of that, what you are looking at. Say, for an example, reverse transcriptase enzymatic activity, you can look at by different methods. And what we have seen here, this is an interesting result, and we have an endogenous retrovirus. And this endogenous retrovirus, it's not a full virus, but it actually produces the reverse transcriptase, and it is integrated in our human genome. We have like more than 40% of our human genome do have retroviral elements. And you can measure that reverse transcriptase, and HERV means human endogenous reverse transcriptase, and that enzymatic activity in different ratios have been measured. So here you can see that there are different ones, 7.1, 10.1, 10.9. These are the different endogenous retroviruses, and you are looking at their RT activity. So you can see that some of these two are the similar, whereas the HERV 10.9 do have a lower activity. It is just showing an overview that there are many different ways that we can measure the viral enzymatic activity, and you can see the enzymatic activity of the different viruses can vary as well. That's another way that you can compare the different viruses. All right, so the next one is the enzyme-linked immunosorbent assay, or ELISA, that you are very familiar with the name. And there are two questions that we can ask through this. Either we detect the viral antigens through this, or antibodies with this. And an extension of this is just your lateral flow test, which is just the dipstick or the saliva test that is being done for the SARS-CoV-2, where you have just a test strip that you have seen. Even for the HIV, that is the initial test strip. Many different things that you do this office test, or a point of care test with your influenza a virus, that you just take a little bit of saliva and you can just do this test very rapidly. So how this is done? So what you have, that in a classic ELISA, which is done in a plate, not a test strip, and we'll come to the test strip in a second, but just on a plastic well plate, you have a solid support, which is a plastic. And here you actually attach an antibody. These are proteins, and these surface are etched, and the antibodies will bind to that. And to that, you can bind a viral antigen, and then you need a second antibody with an indicator, or a fluorophore is attached to that, or a colored reaction, or an enzyme, which will cause a colored reaction. For ELISA, most of the time, this is not a fluorophore, but this is usually an enzyme, like a horseradish peroxidase or something like that, that will cleave a substrate to produce a colored product. A detail of the names of the enzymes are not required, but it will give you a colored reaction. So if you see in an ELISA well that there is colorless, there is no indication of the virus in that well. If you see that there is some color being developed, and these are read by the plate reader, you do not have to rely on your eyes where you see faint color or not, and just this will give you a colored reaction. So how it is done, how this is done in the first place, in this question, so there are two questions being asked. So the first question, suppose you have a patient and you are asking whether the patient does have virus in his saliva or blood or serum. If you're asking that question, that whether you have a viral particle in there, in the body of the patient or not, you will do this assay. So here what you have commercially available, these are the antibodies which will bind to this virus. So suppose you are taking this virus, this is an influenza virus, and this is an antibody that is being raised against the influenza virus, commercially available and attached to a plastic surface of the ELISA plate. You take the saliva of the patient, add to that, and then you add the second antibody, which will also bind to the virus antigen. Both of them will bind to the virus antigen, but the second antibody is added with this enzyme, which will give you a colored reaction. And that will cause a reaction where you can measure that. So here, the question being asked, whether the patient does have virus in the saliva, blood, or body fluid or not. On the other hand, in the indirect method, what you are doing, that you have an attached viral antigen from purified virus attached to the plastic surface. Here we are asking the question, which is being done now, that whether a person has been exposed to the virus or not, whether the person being infected with the virus or not whether the person has virus in its body fluid or not. So if the virus, if the person that or the patient is exposed to the virus, the patient will have antibody against the virus in his serum. So you are taking, or the saliva, you are taking the saliva or the serum from that patient, and you are adding to that. If the patient is positive for that, that antibody will bind to the viral antigen. Now, you add the secondary antibody, which is very different from this one. In the direct ELISA, the second antibody is against the viral antigen. In the second indirect ELISA, this is against the human anti-IgG. Remember that the antibody is an immunoglobulin molecule, and this is an anti-IgG or anti-human IgG molecule, which is usually raised in either rabbit, goat, mice, anywhere other than a human, which is added with a enzyme. And that will give you the colored reaction. So this is done by the ELISA. So just a modification of that, what we called as a test strip test, actually use the same technology where you have a wicking pad and you just have a line of these antigens at a particular place, and you add your sample to one end of that test strip, and by the wicking action of that, the antibody or the viral antigen will be pulled towards the viral antigen across from that. This is exactly like how initially the pregnancy test has been developed. That's the classic test that you all know. And now the same principle works, which is an antigen antibody based lateral flow test with a test strip. It's the same way it works. So that's another way that you can assay. And these are usually rapid tests. But again, you have to be careful. Sometimes the ELISA is great, but the rapid test, particularly the test strip, you may have lots of false positive, particularly when the tests are being done. Say, for an example, for influenza, it is extremely robust and there is very, very less false positive because those have been developed and perfected over many years. For SARS-CoV-2, we are still developing it, and that's the reason that we are getting lots of false positive with these tests as well. PCR is much more reliable compared to that. So you can see that the next comes the viral nucleic acid polymerase chain reaction, and this is used in research, industry, and diagnosis. And the main thing, and you have learned about this PCR, I'm not going to go into this as such, but the overview of that, this Taq DNA polymerase is a thermostable DNA polymerase, which is used in these reactions where you have either the DNA viruses or the RNA viruses. And what you do that you amplify their genetic material and you detect them by some means. And this could be either fluorescently tagged nucleotides can detect it, or classically you run them on a gel and look at the product, basically. So you will use the primers, and these primers are very specific to your virus. So if you are just talking about the different viruses, we have to do a little bit different. So this PCR reaction only amplifies DNA, not RNA. So if you have an RNA virus, you have to do it a little bit different way. For the DNA viruses, direct PCR can analyze the DNA nucleic acid. So what you do for the DNA viruses, you use, suppose I'm just taking an example, that suppose you have a herpes virus, which is the DNA virus. Now you have the primers which are designed to have a particular sequence of some of its particular gene that you already know, and you have the primer based on those genes. So now you isolate the viral nucleic acid, you denature them, and then anneal the primers. And if the sample does contain the DNA of herpes virus, these primers will bind to the specific genes that they are complementary with too. And then you just, again, synthesize the strand, and from one strand, with the help of these primers, it will form two, and then you just cycle them for 35 cycles. You will get billions of copies of that. And if you can detect a PCR product, you will know that the initial sample actually did have the virus. If you don't have the initial sample with the virus, you will not get a PCR product, because the primers or the probes will not anneal to any of the targets. There is no target. So that's how it works. For the RNA viruses, you have to just modify this with a first extra step. And this is being done with the SARS-CoV-2, which is the RT-PCR that you hear every day now. So the RT-PCR, what you do is you use the reverse transcriptase RT followed by the PCR. So what you do that you isolate the RNA from the RNA viruses, and then you do a first step of reaction where the reverse transcriptase convert the RNA into double-stranded DNA. So again, repeating, the RNA is converted to double-stranded DNA. So thus, for the SARS-CoV-2, your RNA will be converted into double-stranded DNA of SARS-CoV-2. So that's that first step you got, and the rest of that you followed by the PCR. So these primers are just basically some specific sequences of the SARS. Say, for an example, you have some specific sequences of the spike protein, nucleotide sequences of the spike protein. And if the SARS-CoV-2 is positive, you will see a PCR product. If you don't, then it is negative. How pure does the sample need to be? Can PCR detect a small amount of viral DNA if there's an overwhelming amount of host DNA? Yes, that's a very good question. Yes, it can. This is extremely precise. And that's the reason that if you have 1,000,000:1 copy, and now the thing is that this technique has been modified over the time to a certain extent, that if you have 1,000,000 copies of other DNA and you have one copy of some of the viral DNA or a bacterial DNA, you can pick up that one DNA. It is that powerful, because after 35 of your PCR cycle, you are only amplifying your own the product that you have only. And so the question is that if you have the host DNA or something like that, how would you know? That's a good question, that how it works. So if you are, say for an example, you have all seen that if you just run a nucleic acid gel and you have all these host DNAs or something like that, and those are higher molecular weight, will probably stay up in the well, whereas you know that where you have your primers from and how long your PCR product should be, because you design the primers a certain way. So your PCR product should be from here to here. And as you design the primers or probe, you know that if you find a product which is 1,500 base pair, and if you get a sharp band at 1,500 base pair, then it is positive. You really don't care anywhere else. You have DNA fragments a little bit, which are contaminated. If you do not have this very specific product, then it is. And how you know that? You will run a negative control where you will not add the primers. You should not have that product in the negative control, whereas in the positive, you should have that specific control of the specific base pair. And this is running the gel, but you can do in the real-time PCR by looking at fluorophore-based probes as well. Does that answer your question, Matthew?
Yeah, thanks. I was just trying to get some clarity about, like Clemson wants us to get a PCR test for
no?
COVID. Yes. Can you hear me?
Okay. All right. Okay. All right. So now here is the controversial thing, and I'm not going to declare war against everybody. You guys, please don't do that. PCR product is not same as the infectious virus. And if I just say that, immediately you was like, then why we are doing RT-PCR? You have to use that data very carefully then. So what is happening that this is an example of the Zika virus, and what you are seeing right here, say 0 days to up to 60 days. If you just look at to take the sample from a patient, or say here from the mouse, you find the infectious Zika virus particle from the mouse for about 3 weeks. But if you take the sample from the mouse, you find the Zika virus RNA for up to 60 days. So this mouse sample, you will be positive for the RNA for 2 months. And that complicates the study. On the other hand, so this is basically shedding from the seminal fluid. So here you can easily see that how it happened, that this male mouse, on the other hand, is vasectomized. And at that time, what you can see that if you have this, that the virus cannot shed into any other parts of the body, you still have a very high Zika virus RNA, not up to 60 days, but still you have a very high RNA. On the other hand, you can see that the infectious Zika virus is very low at this. So here in this vasectomized mice, where you have the virus not being allowed to move anywhere else in the body, pretty much you can see that the detection of the RNA is much less. On the other hand, if you allow the RNA to diffuse through the body, you can see that you can detect RNA after such a long time. So the controversial thing is that, say for SARS-CoV-2, that when you are taking a sample from an environmental sample, say from the wastewater of the Clemson, which is being tested, you are just looking for the SARS-CoV-2 RNA. What does that mean? Does that contain infectious SARS-CoV-2 particle? Absolutely not. So that is the important. So how will you know that the wastewater does have, how will you correlate that data? So if you take the wastewater particle, just pass it through a 0.2 micron filter to take out any other thing, and then you should have the infectious virus particle in your filtrate. And now the only way you will know the infectious virus particle in the wastewater is you have to do an animal cell culture or an animal model and infect them. If you have infectivity, then you know that that wastewater contained infectious virus particle. So the RNA or doing the RT-PCR analysis of that does not give you the infectious virus particle. But that does not mean that data is garbage. What happens that the RNA degrades over the time by the RNase. RNase is a very, very common enzyme. It's everywhere, and the viral RNA will degrade over time. And the amount of RNase in the wastewater is very high. So the RNA RT-PCR analysis from the wastewater can give you an overview of prevalence of the viral disease in the community. So it is, of course, it's an interpretive data. It is not that how many infectious virus particle is in the wastewater, but it is more of a data to understand that how much RNA is being released from the people in that community, because they have to be pretty fresh. That you will not detect the RNAs after a month or so, or even after a week. So pretty much they are fresh coming out from the people. So that's very important. So on the other hand, there are some studies, and these are controversial, that what happened, that there was one patient, and this study came out from China, that they actually found the RT-PCR, this patient was RT-PCR positive for almost 60 or 70 days, one patient. And does that mean that that person is still shedding the virus or not? So usually at that time, that person is not shedding the virus particle as such. The body may have the RNA virus particle. So how we correlate our Zika, our coronavirus samples that is being taken from our nasal swab and doing our PCR. So usually what will happen when we are infected with, say, SARS-CoV-2, when we are losing the infectivity towards the end, after a period, something like that, when the viral titer is going down in our body, we still may have circulating RNA in our body fluid. But you have a fairly good amount. Remember that this is important, and this is a little bit off topic I'm going. You have a huge amount of RNAs, huge amount of RNAs in all your salivary, in your mucus, basically. So your salivary secretion, your nasal secretion, all will have very high RNAs. So if you just have only RNA, naked RNA, which is remnant from the infectious SARS, that will be rapidly degraded in your nose or mouth. So when you are isolating some nasal swab, the RNA must be protected by the coating of the SARS-CoV-2 virus. So the capsid, or the box that we talked about, that is protecting the RNA. And that is why you can isolate the virus from the nose and do an RT-PCR. So that's the reason that it is very, very useful. The study that I talked about, that after 60 days or 70 days that SARS-CoV-2 RNA was available, that sample was taken from the blood, but not from the nose or from the saliva. So that's some of the detailed idea that how these data can be used. All right, so after that, we are looking at some of the viral reproduction, the burst concept. And these studies actually started in 1937 by Max Delbruck and his colleagues, who were just talking about, were working with a T-even bacteriophage. T-even means T2, T4, T6, and so on. So the burst concept, so what they did, that the yield of a virus particle from one cell in one viral generation is called the burst, because a viruses literally burst out of the infected cell. So what happens that you just allow the bacteriophages to absorb the the virus, and for some time the viruses actually completely disappear, and then suddenly, just as a burst, they come out. And we talked about that a little bit. So for example, you can just produce approximately about 150 bacteriophage per bacterial cell. That's the most that they will allow. On the other hand, eukaryotic cells being much bigger than the bacterial cell, you can have up to, so this number will be 10,000 per eukaryotic cell. But the burst is not observed in many viruses. They shed viruses by budding, and we could talk about this for a while. So what they did, that what are the different types of things, that the first, how the Max Delbruck actually studied, Ellis and Delbruck, that E. coli bacteriophages, they will take the E. coli cell and they will add the bacteriophages to them. And they will allow it to absorb, which means the bacteriophage will lodge onto their cell wall for a very short period of time. And immediately after that, they will dilute the culture. Now, remember that in this experiment, short period of absorb, then immediately diluting the culture, this is the most crucial step out of all of them, because you actually try to infect all the bacteriophage at the same time, which we call as a synchronous infection. If you don't dilute the culture, then you may have, over the period of time, you may have other bacteriophage trying to infect the E. coli as well. So that's why you dilute the culture. And then these cultures, that after dilution, you take the sample from time points. So that means that from the diluted culture, you take samples from 5 minutes, say every minute, like 5, 6, 7, 8, 9, 10, 11, 10, 11 minutes. And you sample, and taking the sample, and then you assay that how much bacteriophage that you have in that sample. And what they found for that we're going to see that what they found, that right here, they actually, after absorbing, they dilute the sample right here. After that, they are taking the sample from different time points, which is not shown, but you can see that this is a time scale. So it looks like that from the sample, the virus actually disappeared for such a certain time, which is called the eclipse period. And after that, you have a burst or of viruses, basically, and then it will plateau out. So what we mean by this at this point, that after this eclipse period, all the viruses actually almost come out in a synchronous fashion out of all these cells. And this is what is called a burst. And this is what we call as a one-step growth cycle. Unlike, if you just look at, we talked about that, unlike the bacterial growth curve, where you can see that where the 1 divides into 2, 2 into 3, even in the exponential growth, you see a gradual increase in the number of cells before it plateaus out. And this is a typical reproduction in the bacteria. And we already talked about that why we see this eclipse period and the burst, because the virus particles are made from a preformed viral particle, and they are coming out after being assembled. So in here, those components of the virus are being made, then assembled, and they are coming out all the time. Now, here is an important thing that we are looking at, which is the extension beyond these curves that we have seen before. In this, all cells are infected, which means that we did a synchronous infection. If you take 1 million cell or E. coli, you have infected theoretically each one of them with the bacteriophage. You will see a one-step growth curve. On the other hand, what you do that you can have a multi-step growth curve. If you say instead of 1 million cells, out of that, suppose you 100 or 1,000 or 10,000 cells are infected, you will see multiple burst phases. And what we mean by that, some of the cells will make the viruses. These viruses will come out and infect some other, and you will see that multiple step. And again, you will see that there is a possibility of the viruses present from the previous one, and that's the reason. Also, the number of cells will be lower because some of the cells are already been killed, and that's why it becomes a little bit more curved rather than a sharp rise in there. So few cells infected, you will see a multi-step growth curve. This gives you an overview of that, some very interesting concept. So here, what we are looking at, that on the left-hand side is an adenovirus type 5, which is an extracellular, which is a naked virus. And this actually assemble, and the cell will burst, and it will come out. And we'll just come back to the B later. So here, there is a little bit of problem with this. This intracellular is incorrect. So here, this is the extracellular virus. The red is the intracellular virus, and the purple, this should be the total, intracellular plus extracellular. So this is a problem with the book itself, which will be total amount, but really doesn't matter. Intracellular and extracellular viruses. So how this experiment is being done? You take some of the specific cells that you infect with the adenovirus type 5, and you add the viruses, and you infect the viruses in a way that all the cells are infected synchronously. And we will learn that how you can do that. So, so I did not. Are you guys okay with the PowerPoint slide as such? Everybody? Okay. Sorry, I just read that a little before. Anyway, okay. So synchronous infection, and you are taking here hours after the viral absorption because these are much slower. See, for example, for the E. coli and the bacteriophage, it is a matter of minutes that how fast they actually assemble, whereas all different viruses will have different rates. So here you can see that initially when you diluted the virus, at that time, still you have extracellular virus remaining from the sample that you have added to the cell. That's why you have a little bit higher amount of virus. And then gradually those viruses will enter into the cell and they will disappear. And you have the eclipse period right here. You don't see any extracellular virus at this point of time. And then what you see that you will see a gradual increase of the intracellular virus right here, right, which we call as a burst. But you will not see the extracellular virus still certain time. So there is a latent period which extends beyond the eclipse period. And why this is so? And this is a very interesting study that how you do this, you sample the infected cells, and then you do two things. For the extracellular virus, you will just take, first of all, after taking these, scraping the cells out of your flask or the Petri plates, you centrifuge them down. So if you centrifuge them down, the pellet will have the cells itself containing the virus, supposedly. And the supernatant will have the virus which is outside the cell. So you can see that right here, extracellular virus, and these are the intracellular virus, and the purple is the total virus, total of these. So what you are seeing that right here up to this, in the eclipse, after the eclipse period, your intracellular virus, which is found from the pellet of the cell, is increasing. So what we mean by this, that at the end of the eclipse period, the virus is started assembling, and the amount of virus inside the cell is increasing right here in this portion, in between this eclipse and the latent period. But we are not finding any virus in the extracellular fluid yet at the end of the eclipse period. So here it's a beautiful experiment where you can see that just this difference from the eclipse period to latent period, this part is where the virus is being assembled inside the cell, but they have not come out of the cell yet. At the end of the latent period, the virus will start emerging, burst out of the cell. And that actually shows this type of virus, where in the adenovirus type 2, which actually kill the cell and burst the cell, and they all come out. Whereas here, you actually see a very different way. The Western equine encephalitis virus is a budding virus where they will not burst the cell, but they will gradually emerge from the plasma membrane, and they will keep the cells alive. And this is a gradual process rather than bursting what you see here. So that's the reason that what you are seeing, that after initial infection, you are seeing that the intracellular virus is actually rising and then kind of plateauing. And then the extracellular virus, you don't see anything initially. So this is your eclipse period right here. On the other hand, this is your late eclipse of the latent period. You don't see anything. On the other hand, extracellular virus after that also is coming out gradually and plateauing over the period of time. So you can see that what is happening right here, difference in between that your intracellular virus, as they are being made, they are going to the plasma membrane and they are maturing in the plasma membrane. So again, this is the important part. Here, the virus is completely matured inside the cell as a full virus particle. They are infectious inside the cell. They burst the cell, come out. Here, the viruses are not maturing into an infectious virus particle until they come out or bud from the plasma membrane. And that's the reason that you do not see the intracellular virus amount very much, but you see the extracellular virus. So these are the two different ones, that this is a budding virus which mature at the plasma membrane versus a virus which actually make the complete virus inside the cell and burst out of the cell. All right. So to do this, basically, what we are looking at, the synchronous infection that we talked about, that how we do this synchronous infection. All these experiments need a synchronous infection right here, particularly for the one-step growth curve that we have seen. So here, there is a new term that we are talking about, is the multiplicity of infection. The number of infectious particle added per cell, or plaque forming unit over number of cells, not the number of infectious particle each cell receives. So for an example, that if you add 10 to the power 7 virus particle to 10 to the power 6 or million cells, so means you add 10 million virus particle to million virus particles. So then the multiplicity of infection of 10. That means that theoretically you will think that each cell should get 10 virus particles, but it will not. The reason, we are just going to explain that, why the MOI is important, or the multiplicity important, and the implications. So this is the experiment. The infections depends on the random collision of virions on the cells. When susceptible cells are mixed with the virus, some cells are infected, some receive 2, some 3, and more particles. And this distribution particles per cell is best described, which is known as the Poisson distribution. So by the Poisson distribution, just think about that you just have this bag full of this tennis balls, and you have these buckets, and all these balls, you just throw them randomly on top of these. Some of them may not receive any, some of them maybe 1, 2, 3, and there will be a random distribution of this. And statistically, you can actually find that how you actually can calculate this. So again, this is not needed. The math part is not needed, but just for your reference, that how you calculate this is the Poisson distribution. So here, this pk is e to the power minus mk over k factorial. So here, the pk is the fraction of the cell infected by k virus particles. And this m is the multiplicity of infection, or MOI. So we'll just break it down in a very easy way. And again, remember that I just said for reference only, not needed for the quiz or exam. But in a virology course, we definitely need to understand that how we come to this number, or the MOI. That's why we are just talking about this. So if we have uninfected cells, then the P number, the K, remember that that's the fraction of the cell. This is 0, is e to the power minus m. Cells receiving 1 particle, P1, equal to m e to the power minus m. And remember that m is the multiplicity of the infection. On the other hand, cells with multiply in multiply infected, that means that cells, if they receive more than 1 particle, this will be 1 minus e to the power m plus 1. So this is obtained by subtracting from 1 the sum of all probabilities for the value of k and the probabilities of P0 and P1, P0 and P1. So that's basically that how you can actually identify this. And MOI is denoted by the lambda, which is called an event rate, and this E is the Euler number. So if you just plug in these values, and it looks complicated, but it is not that complicated. If you plug in these numbers, it's very easy. And if we just look at an example, it's even easier. So let's take a look at this. So here, if we take 10 to the power 6 cells that are infected with at 10 MOI of 10, or multiplicity of infection of 10, 45 cells are uninfected, and this we get from the Poisson distribution, from that equation. 45 cells are uninfected, and about 450 cells receive 1 particle. Rest receive greater than 1 particle. So this is a very interesting observation. So you can see that out of 1 million cell, pretty much, except 45 cell out of a million, all of them will receive at least 1 or more than 1. So at MOI of 10. If we look at an MOI of 1, 37% cells are uninfected. 37% cells will receive 1 particle, and 26% will receive greater than 1 particle. If we take an MOI of 0.001, 99.9% of the cells are uninfected. So that's how it goes. So why this is so important and where we use this. So to have a synchronous infection and to study a single burst growth curve when we are assaying, we will actually infect the cell at a particular MOI. So if you just want to, so here is a question, who can answer this? We'll see. So here I'm asking the question that if you want to infect the cells synchronously for an experiment, which is a one-step growth curve that you have seen already before, which will be like this, one-step growth curve. What kind of MOI you are going to use for a one-step growth curve? Are you going to use a high MOI, or are you going to use a low MOI for a synchronous infection? Anyone? Either you can type or you can talk. High or low? How many people say for a synchronous infection you need a high MOI? Just Matthew? Anyone else support Matthew? High MOI for synchronous infection? That is correct, actually. Yes. So because see what happens, your goal for a synchronous infection means out of the million cell, you want all of the cells to be infected. And you can see that out of a million cells, except 45, almost all of them are infected. This is synchronous infection. And this will produce a single step growth curve. If you infect the cells with an MOI of 1 or 0.001, what kind of growth curve you will get? Anyone? You can either type or talk. We talked about that. So here you will get the multi-step growth curve. So that we talked before, right here, you can see that a small number of cell is infected in that. So that few cells infected. So when we are just used a low MOI, only a few cells will be infected, and we will get the multi-step growth curve where we use a high MOI to infect the cells. All of them will be infected at the same time synchronously, and we will just get a single burst or a single step growth curve. And this is the take-home message from that when we are studying the viruses for our study, for a single step growth curve, we use a high MOI. This will be your question for your quiz or something like that, that for a single step growth curve, what kind of MOI you use? Like 10, 100, 1,000, you can use 10,000 even, just to make sure that every virus receives the particle. And now remember that the PFU particle ratio will complicate this study. If you just use something, a virus does have a particle to PFU ratio is about 10, you want to use them at least 10 or more. But if you have a particle to PFU ratio is 10,000 like a papillomavirus, if you're using just 10 MOI, that is not enough to infect to the 10 to the power 6 particles, because most of them are defective. They're not getting. So here is the question again, we are just talking about back again, that if you're having the particle PFU ratio is very high, you have to infect the cells even with a higher MOI than calculated MOI. Whereas if your particle to PFU ratio is low, your calculated MOI, you can be infecting the cells close to the calculated MOI. If you want a multi-step growth curve, you can use a low MOI like a 1 or 0.01 or 0.001. And that's pretty much all of it, I guess.